I'm Robert Dillman, a medical oncologist and cancer researcher in California. For more than 30 years, I've been involved in the development of cancer biological therapies and immunotherapies, including monoclonal antibodies, lymphocyte therapies, and cancer vaccines. Dove Press asked me to write a review covering melanoma vaccines because of the great interest in this approach. A comprehensive review entitled Melanoma Vaccines, Trials and Tribulations is now available in the online journal Vaccine, Development and Therapy. We know melanoma can respond to immune therapies. 3 immunotherapy products are already approved to treat melanoma, and others are under development and or about to be approved. This review covers many variables associated with melanoma vaccines, including adjuvants and co-stimulatory agents, sources of melanoma-associated antigens, and the results of clinical trials, which are summarized in a series of tables. Many vaccines are given with co-stimulatory agents called adjuvants. The various classical adjuvants that have been used as components of melanoma vaccines are reviewed, as well as clinical trials of melanoma vaccines that have employed cytokines, including alpha and gamma interferon, interleukin-2, and GM-CSF. Recognition of the importance of dendritic cells for antigen presentation has led to a number of trials testing anti-melanoma dendritic cell products using peptides or tumor cells as sources of antigen. There are many possible sources for melanoma-associated antigens, including specific molecules recognized by monoclonal antibodies and or cytotoxic T-lymphocytes, allogeneic tumor cell lines, and autologous tumor, either as manipulations of whole tumor or as autologous tumor cell lines. Clinical trials using each of these approaches are tabulated, as well as approaches involving injections directly into tumor in vivo. The identification and characterization of melanoma tumor stem cells has focused attention on vaccines that may target such cells. Although randomized trials with allogeneic cell lines have been disappointing, autologous tumor cell line approaches are now feasible. Trials with such patient-specific autologous tumor stem cell products, especially when combined with dendritic cells and GM-CSF, are encouraging enough to justify confirmatory randomized trials in patients with metastatic melanoma. The summary includes my opinions, based on the current published evidence, regarding adjuvants, dendritic cells, monovalent versus polyvalent vaccines, allogeneic versus autologous sources of antigens, and whole autologous tumor versus autologous tumor cell lines as antigen sources, and how melanoma vaccines may enhance the benefit of monoclonal antibodies that target immune checkpoint molecules. I think you will find this an informative and comprehensive review. Thank you for your attention.